taking a look at your paper models, what did you notice? Are both of them upside down? Okay, so one of them is upside down. So the initial chain we labeled together on Friday. The top, was that 5' or 3'? Of the first original chain that you had. There was 5', and then the bottom was 3' in response. So the opposite chain is the top 5' or 3? It's 3, and the bottom of it is 5. What do we call that? That's called being anti-parallel. And in our initial pictures, like the one from your textbook, it tried to explain it to you why it was anti-parallel. But I think in the textbook, it's confusing to see that they run opposite directions because in the book, all the letters are going the same way, right? But on your paper, it's obvious because one of them is upside down. Yep. Yep, that's exactly what that means. And it doesn't seem like when we first talked about it, when we were talking about vectors and things going one direction and another in 3 dimensions in space, it doesn't seem really obvious why it has to happen until you take a look at the piece of paper in front of you. If the nitrogen bases are going to be touching each other like they should be, if they're going to be next to each other like they are in reality, then one of them has to be upside down, right? That's the only way we can have the nitrogen bases occur next to each other. They wouldn't be paired in a complementary fashion. So we've noticed that it's anti-parallel. What do you notice about the number of adenines and thymines? So the number of adenines equals the number of thymines. And what about the number of cytosines and guanines? The number of cytosines equals the number of guanines. This is actually a well-accepted rule named for the scientist who discovered it. We call that Chargaff's rule. He's the guy, I believe his first name was Edwin. I think Edwin sounds right. He's the guy who said, well, the number of adenines equals the number of thymines, so they must be complementary. They must pair together. The number of guanines equals the number of cytosines. This pairing is actually really very important. Very, very important. Shrink that a little. Shrink that a little. Move those out of my way so I can make this a bit bigger for you. So the reason that it becomes so important is what holds the nitrogen bases together in the middle of the strand is drawn in red right here as red dashed lines. Those red dashed lines are a type of bond that we learned about in Chapter 2 called a hydrogen bond. And it occurs between an element that really loves electrons and hydrogen on another molecule. So it occurs between an element that really loves electrons, like oxygen and nitrogen, they love electrons, and hydrogen on a molecule next to it. You wouldn't have oxygen and hydrogen or nitrogen and hydrogen or oxygen and hydrogen line up if you violated these base pair rules. They wouldn't match. So no hydrogen bonds could form to hold our nucleotides together. It occurs between an element that really loves electrons. We call that electronegative. So an element that really loves electrons and hydrogen on a nearby molecule. If I paired guanine with adenine, yes, we would have an oxygen up at the top that lined up with a hydrogen. But then we would have nitrogen next to a hydrogen. I don't know if that's going to work in 3 dimensions. And then down at the bottom, we'd have a hydrogen next to a hydrogen. That can't form the bond that's needed, so they can't improperly pair. Without these bonds, the DNA molecule can't stay together. So they have to pair that way. It's absolutely important. And if they weren't anti-parallel, if they weren't upside down in respect to each other, then these wouldn't line up perfectly to make bonds. It's kind of an important part of it. We already said they're anti-parallel, and if we look here, we've got our 5 and 3, and our 5 and our 3. On the sides of your molecules, you'll notice you should have sugars and phosphates alternating, all stacked up on top of each other. Do you have that? How many of those do you have? Sugars and phosphates all stacked up. Mm-hmm. So you should have like a phosphate and then a sugar and then a phosphate and then a sugar and then a phosphate and then a sugar. How many of those strands of that stacked on top of each other do you have? Not how many sugars and phosphates. How many, like? 2. You should have one on one side and one on the other. Do you see them clearly on your molecule? We call that the sugar-phosphate backbone. So there's 2 sugar-phosphate backbones. And then across the center of your sugar-phosphate backbone, you have nitrogenous bases. Now that I've colored over this, does it look more like the DNA molecule you're accustomed to seeing? Like a ladder? What you have in front of you on your paper is what it really looks like. Now, there is one more thing. God bless you. Emily?
What's across the center of the sugar-phosphate backbone?
What's across the center of that sugar-phosphate backbone? What are A, T, C, and G? Nitrogen. Nitrogen bases. I just drew all over this picture.
So you just keep the phosphate and the house like on the outside, but then and then everything else is like towards the center.
How many nitrogen rings occur in every step of that ladder? 1, 2, 3. Is that true for every step of the ladder on your piece of paper? So the number of nitrogen bases across the center is always 2 bases, 3 rings wide. What happens if I accidentally paired adenine with a guanine? How many rings across would it be? 4. What if I accidentally paired a cytosine with a thymine? How many rings across would it be? 2. Have you ever seen a ladder that looks like that? Pull down your attic ladder. Does it look like that? Roofers, when they lean a ladder up against your house to put a new roof on. Does it ever look like that? They're perfectly straight lines on the side with an even length of rung. That's true in DNA as well. So base pairing rules matter. The base pairing rules are going to matter. Anything else we should be able to observe about your structure? A always pairs with T, C pairs with G. There's a certain number of hydrogen bonds across the middle. They're anti-parallel, so one looks upside down. Anything else about it that's... are the two chains across from each other the same length? They should always be the same length. What you have in front of you is only 8 nucleotides big. Your whole genome contains 3 billion base pairs, so 6 billion nitrogen bases. So every cell you have has 3 billion pairs of those in them. A genome is every nucleotide that an organism has in order. That last thing's kind of a big deal. That in order thing. Some of you, when I told you to add 4 more bases on, I was like, just add 4 more bases so it's a total length of 8 bases long. I don't care what order you put them in. Doesn't matter. That's because there's not a specific order they go in. The order they go in changes for every single organism you look at. Not only does it change for every single organism you look at, quite frankly, it changes for every individual that you look at. We all have the same genes, but we have different versions of those genes. So our genes are made up of nucleotide bases, right? We all have the same ones, like we all have a gene that turns our pee pink when we eat beets. So we all have that gene. It'll tell you whether or not you've eaten beets. It's whether or not you can process that dye and break it down. If you can't break down the dye in beets, then you pee it out pink. And if you can digest it, then your pee is pea color. You eat a lot of beets and you've never noticed it before, you likely have the enzyme that helps you digest that dye. The difference, then, is the order of nucleotides that you got. So those of you who don't have pink pea after you eat beets got A, T, C, G, G, C. And those of you who do got A, T, C, G, G, G. Just a difference of like a couple of nucleotides in the order they're in.
So every person has one genome that's the same throughout their entire body. It'll never change, like no matter what.
Every single cell you have has the exact same genome in it. The only time that it changes is if there's a mutation, and those mutations tend to lead to diseases like cancer, which is why cancer is localized, because the genome of that one cell has changed.
Does everyone have a different genome?
We all have a different genome in that all of us have different nucleotides, right? We all have different, that's why we look different. But we all have the same set of genes, but different genomes.
So every cell has how many?
6 billion nucleotides, 3 billion pairs.
So is that like what causes allergies and stuff, like a different order?
That's what makes every single thing that you can think of about us that's different, that's what makes it different, is the order of nucleotides that you got. And when I say every single thing that makes us different, I mean whether you have blonde hair or brown hair, whether you have blue eyes or green eyes, whether you can or you can't.
Wait, I can't do that.
If you can't do that, it's because you have the gene that prevents your tongue from rolling like that. Why though?
I feel so ugly.
I don't say like the type of person.
Whether or not you have a widow's peak, whether or not you have a cleft chin. Don't worry, you'll never be able to do it. It's not worth trying.
I'm gonna train myself.
Good luck with that. It won't change. The only thing that makes us different then is the order of our nucleotides. That's our genome. But here's the trick. you have, you all started as one cell, right? Like that's how this thing works. You all started as one cell, and that cell divided in half had to make a perfect copy. And those cells divided in half and had to make a perfect copy. There has to be a quick, efficient, and accurate method of copying 6 billion things. How many of you are like, psh, sign me up, I can copy a 6 billion page book with no mistakes? No? Every single one of your cells in your body has the ability to do so. It copies 6 billion things with 100% accuracy. And if it does make a mistake, it's got a built-in proofreader that goes back and fixes it.
So if there's something that's wrong with one of your nucleus, your cells are going to have that problem with that.
If it is the first cell that you were made out of.
How does a cell just accumulate a problem like that?
There are a bunch of different ways. It could have inherited it from the ovum or sperm that it was made out of. It could have just gotten a bad one. Or it happens because of the environment. So you were exposed to a chemical and it caused a change.
Wouldn't that be like in the first cell? Like what if you what if you have like, I don't know, just, this is really random, like 8 cells and then one of them's bad? How does that become bad?
There is a reason why they put a giant lead vest on you when you have x-rays at the dentist, right? You ever wonder why?
No?
I just went to the dentist yesterday.
You know why they put that big lead vest on you?
No.
Because x-rays have the ability to mutate your DNA. So they cover all of your vital organs.
Oh, that is sick. Wait, that's actually bad at the same time.
Yes!
So, like when they take x-rays, they put the lead, but does the rest of your body still?
It tends to focus it in a very small area. And it's such a low dosage that the improbability is, it's improbable. However, there's a reason why you only get x-rays every so many times you go. And there's a reason that your dentist or healthcare practitioner leaves the room to push the button. You ever wonder that? You ever like, oh, they put this thing up to my face and then they walk out?
Yeah.
Wait, why do they walk out?
Because they can't be exposed to that much x-ray over and over again all day. Ah, now you're skeptical. You're like, don't get near me with that.
Like, what does it do? That's dangerous.
Did you just miss this conversation?
I kind of zoned out in the middle. But like,
It causes diseases. It causes DNA to mutate. Same reason you wear sunscreen, right?
I have
very funny.
So we have to make sure that there's a system that is 100% accurate. Every cell has that ability during interphase of the cell cycle. During S, that's when all 6 billion get copied. That's why we do this chapter after we did the cell cycle chapter, so you know when this happens in the life of a cell. It happens during S. Mm-hmm. All of the DNA is copied during synthesis. Yep. Mm-hmm. So every single nucleotide gets copied during that S phase. And think about, really, S was a significant portion of the wedge, but it wasn't half of the wedge, was it? Was it 75% of the wedge? What about 33 and a third? About a quarter to a third of the wedge was S. That tells you how incredibly efficient this process is. The process during synthesis is called DNA synthesis or replication. The process is semi-conservative. What does semi mean? And if I am trying to conserve water or conserve energy or conserve resources, what am I trying to do?
Save it.
Trying to save it. So semi-conservative means half saved, half old. The process, or we discovered that DNA synthesis is semi-conservative through an experiment. It's pretty famous. It's called the Michelson-Stahl experiment for the 2 scientists who did it. They used bacteria. We're going to watch a short video about how that worked. I'll just tell you that this process is different in prokaryotes and eukaryotes. You are about to see it in a prokaryote. So you'll see the process, or at least they'll talk about it, in a bacteria, prokaryotic cells. We'll talk about some of the finer differences between proks and euks, but just so you know before we start. Just to give you a little background for Messelson-Stahl, when your DNA replicates during synthesis, it has to get the new nitrogen bases from somewhere, right? Like, how do you build a house if you don't go somewhere and get bricks? Do you agree? You have to find bricks somewhere? Yeah. It's not like you just went in your backyard and magically there was a pile of bricks there. Was there? Is that how that worked? You get them, like you buy them from elsewhere. Your cells have to take nitrogen. Mm-hmm. Question?
How do you spell the
STAHL. So your cells have to get the nitrogen from somewhere. It's going to get it from the environment. That's just that'll help explain why Messelson-Stahl chose to do what they did, knowing bacteria had to get the nitrogen for their nitrogen bases somewhere. When we say the term semi-conservative, supported by the Messelson-Stahl experiment, that means that when DNA replication occurs, we have two daughter strands of DNA, where red is the original and green is the new strand. So of the two daughter strands that are made, half is old and half is new. This process, yep. The semi-conservative means when two daughter strands are made, one half will be from the old original strand, and one half will be from the new strand. Mm-hmm. Mm-hmm. One strand in each of the copies, yeah. This seems like something that you're like, yeah, that's not really that really very important, except understanding this was absolutely essential to our understanding of how replication worked. Figuring this out meant that we could posit a hypothesis for how replication occurs. This process is different in prokaryotes and eukaryotes. I should tell you, in prokaryotes, there is one spot that it starts. So in a proc, there's one spot that starts, and in a euc, there's why. In a prokaryote, there is one single circular chromosome. That's all any prokaryotic cell has, is one single circular chromosome. And it's really that simple. You memorized that for your table where you told me the differences between prokaryotes and eukaryotes. If replication began right here then, at what we call the origin of replication, and it worked its way around making a copy, it eventually ends where it started, right? It's a circle. In eukaryotes, not only is it not a circle, they're linear chromosomes, but there's multiple. How many do you have as a human? How many chromosomes? "23 pairs for a total of 23 pairs makes 46 total. So in every one of your cells, you have 46 different chromosomes. If there were only one place that replication occurred, say on chromosome 1, it could work its way to the end, but then chromosome 2 would never get replicated because it's a separate linear strand. Nor would chromosome 3, 4, 5, 6, all the way up to 23. You'd never have all of your DNA. So in a eukaryote, replication begins in multiple places. So there are multiple origins of replication. In a prokaryote, there's one, and that's it. When the origin of replication opens, it's going to open and move in two directions. That origin opening and moving in two directions is going to create what we call a replication bubble. And if I cut that bubble in half, if I only look at half of it, it makes a replication fork. So for every origin of replication, there's one replication bubble and two replication forks. Here's the bubble. And there's one fork on this side, and then one fork on that side. That's at every origin that happens because it moves both ways. Moving both ways lets it happen faster. So on that prokaryotic chromosome, the circular one we saw, it would start in one spot, move both ways around, so it would meet halfway on the other side. It'd be twice as fast. I know it's a lot of words. I'll let you copy all those words. Yep, so this is the before replication starts. This is when it starts. We are going to go through all of the major players in replication. They all have special names. And as we go through them, I'll tell you we're going to watch a video of it all in motion after we learn the major components. It's going to be a little bit confusing at first, but I promise with the video when it's in motion, it makes a lot more sense. Yep.
I'm so very confused by the semi-conservative, like, DNA synthesis. Like, I don't get it. Like,
When we start the replication, I'm going to use different colors, and it might then become more obvious. Once we see a strand in motion, if it's not obvious or I don't point it out again, raise your hand and say, on this picture, will you show me why it's semi-conservative? Here are all of your major players. I'm going to have you write them down before I tell you what any of them do. It involves a whole bunch of enzymes. Anytime we see the letters ASE on the end of the word, we know it's an enzyme. An enzyme is just a chemical that speeds up a chemical reaction without becoming part of the product. So they aren't used. They make it happen, but they never get used. Wow, that's really messy. Hold on. Torsional stress. Torsion just refers to twisting. So there's a certain amount of stress or pressure applied to something when you twist it. Same thing happens if those of you who crack your backs by twisting your whole body, you get that torsional stress on your body. Your body uncurls, right? It doesn't stay curled up like that. If it did, we'd have a serious problem. Now all of you are like, I feel the need to stretch my back like that. Go for it. Why not? You've been in chairs all day. No judgment here, friends. You'll notice that I am infrequently in the seated position. So that's number one player. They come along, they relieve the torsional stress. Number two player is our friend helicase. Yesterday we looked at how the nitrogen bases are held together by hydrogen bonds. And in order to cut the hydrogen bond, you have to have an enzyme to do it, because the hydrogen bonds like to form. They're like magnets that are attracted to each other. Get a magnet close enough to another magnet, they stick together. That's what happens with the hydrogen bonds. You get two nitrogen bases close enough, they get stuck together. The only way to unstick the magnets is by physically applying a force. The only way to break hydrogen bonds is physically applying a force. Who does that? The enzyme named helicase. That's its job.
What's the SSBP?
We're going to get there. So it breaks those hydrogen bonds. Think about it this way. Consider your DNA like a zipper on your backpack. You have 2 zippers on your backpack, right? So when it's all zipped up, it's at the top. Helicase is like the zipper pull. You pull them in opposite directions and it pops the teeth of the zippers open. Exactly. So its job is going to be to cut the strand in half, but just like magnets, if you separated the magnets but left them close enough together, what are they going to do? They immediately go back together. If you didn't apply what we call single-stranded binding proteins, we stick them on the outside of the helix and it prevents the hydrogen bonds from reforming. It leaves it open. If we consider our DNA just a whole bunch of recipes like a cookbook, just like a real cookbook, you can't copy a recipe if the book doesn't open. So we're opening the book here and leaving it open for the process of copying. You guys ever painted a wall before? You had to use primer when you painted the wall? Why do we put primer on walls? Make sure the color goes on smoothly, because if you don't put it on the wall, especially if you use a vibrant or dark color, something that's very colorful, I don't mean like beige, which everyone apparently paints their walls like beige or eggshell or white or cream or something neutral. I mean like you've gone crazy and you decided your wall is going to be navy blue. You got to put some primer on that wall. If you don't, the pigment has nothing to stick to. That's the point of primer, is to provide pigment something to stick to. DNA can't stick to nothing. There has to be a primer attached. So that primer that gets attached is attached by the enzyme primase. So primase attaches a primer of RNA. So it's not a DNA primer, it's an RNA primer. You'll notice helicase and primase. Their names are related to their functions. Helicase unwinds the helix, where primase attaches a primer. So we take its function and we just add ACE to the end.
So RNA is the primer, but primase is the thing that attaches it to the strands.
Exactly. Precisely. It acts as like a temporary placeholder, so then we can attach other DNA to it. You'll see where it fits. When I draw out the picture, you'll see where it fits. As we do this, I will say there's only one more enzyme. One more. DNA polymerase. Remember, we just take its function and add ACE to the end. The function of DNA polymerase is to make a polymer of DNA. That's all that DNA is, right? It's a polymer. It's one giant molecule. So it attaches complementary nucleotides. I will say DNA polymerase has one quirk. I don't want to call it a flaw. Flaw sounds so judgmental. It's got a little quirk. When you read, what direction do you read on your piece of paper when you're reading in English? Left to right. If you try and read from right to left in English, there are no words on the page, right? Like it's a string of unintelligible letters. Doesn't work. DNA polymerase is much like us. It can read the template strand in only one direction. So it's going to read the template from 3' to 5'. So it can only go one way. It's a quirk more than a flaw. We don't blame it. It changes how we do things a little bit, but you change all sorts of things you do for other people's quirks. Now we'll see how all of these pieces fit together. I'm only going to draw you one replication fork. How many are there at every origin of replication? Excellent. The top strand is 3' to 5'. What's the bottom strand? Let's see. Now that I've used pink, let's go with yellow. First thing that happens, topoisomerase, we're going to give it a diamond shape, is going to attach at the origin of replication, proceed down the origin to release any stress or pressure. We'll label topoisomerase with a nice T. Second thing that happens, helicase moves down the strand and pops open all of those hydrogen bonds. Our friend primase is going to move in and attach an RNA primer at the origin of replication on the 3' to 5' strand.
Does it always start at the 3 to 5?
Because that's the only direction it can read. So it attaches a short primer.
Is it the DNA tolerance?
It happens for both of them. They can both only go in that direction. So here's our primer. It's been attached. Then primase actually floats away. It attaches the primer, and then it falls off. Our last enzyme, DNA polymerase, what color haven't we used yet? Blue. DNA polymerase attaches and it's going to attach complementary base pairs to the strand, moving on the template from 3' to 5'. So say the first thing that DNA polymerase encounters is an adenine. What is it going to attach? It attaches a thymine. So I'll erase those because I don't like the way it makes my drawing look. So it's going to attach a thymine because it encountered an adenine. And then it reads the next one. It finds cytosine. What will it attach? It's going to attach guanine. And it's just going to proceed moving that direction. So it's going to have been made in the previous part of interphase. So it takes all the nitrogen from the environment and constructs them. Yep, that's where, so DNA polymerase has something to stick a DNA nucleotide onto because it can't stick to nothing. It's what brings the complementary nucleotides. So these are nucleotides, because the original nucleotides this paired with are over here, right? So there's nothing over here to stick onto it. So primase attaches our small section of primer, and then hydrogen bonds are naturally going to form between those bonds, or between those nitrogen bases. It's going to get replaced at the end of the. Yep. So it's a placeholder. Everything just continues down the strand. This keeps going that way. Typoisomerase keeps going that way. Everybody just keeps going. Oh. 5' on the bottom is just a little bit more complicated. Just a little. Do we think we're okay with the 3 to 5 on top first? We're like, all right, sure. You unzip it and then you attach the complementary things.
I think I just got the semi-conservative. Is that where the one old strand of DNA comes from and the other one?
You got it. I told you, when you see the picture, you're like, yeah. That's why I use 2 different colors, right? Because now you know, here's my new strand, there's my old strand, because we use the old one as a template.
I'm sometimes confused on what the 3 and 5 are.
That's the direction that your sugar is facing. Remember, oxygen on top or oxygen on the bottom? So on this strand, my oxygen is facing that way. And on this strand, oxygen's facing that way. DNA polymerase can only read it when oxygen's facing that way.
Oh,
So that's the only direction it can read. This top strand, this process is very quick. Very, very quick. So we call it the leading strand. It's leading the way, right? Joe's question was a natural one. You're like, well, we talked about this one and it keeps going, but what the heck about the guy on the bottom? He lags behind a little. So first we look at the leading strand. We also call that continuous. We call it continuous because it just is going to continue, and it keeps going, and it keeps going, and it keeps going until it reaches the end. No big deal. Down here, it's quite inconvenient. This other strand down here, primase can't attach a primer here and read in this direction because it's 5 to 3 on my template. Can't go that way. But my strand is going to keep opening that way. So instead what happens is primase is going to attach right here. And it's going to add our lovely little RNA primer. Then we'll have a different DNA polymerase who shows up and attaches complementary nucleotides and moves that way. How many of you are like, well, that's not a problem. This doesn't seem complicated. It's just going to move the opposite direction. Except what's helicase doing? What way is it going? It's going to keep going this way, right? So what about all the new DNA that gets opened?
Another primate.
Another primase attaches and works back until it hits this one. So we'll do another complex of this exact same thing. So the bottom strand has to be made in pieces, and the pieces have kind of a funny name. They are named for the scientist who discovered that this happens. They're called Okazaki fragments. Naturally, because this happens in pieces, this process is ridiculously slow. Rather than leading the way, it lags behind, so we call it the lagging strand. That lagging strand is discontinuous because it's made in pieces, in very tiny chunks. We're going to see some Okazaki fragments being made in a video that'll make it more obvious because helicase will actually be able to move. we'll watch it through once with the narrator, once at our own speed. The last thing that's going to happen here for this whole thing to finish up, our primer is going to be replaced by DNA polymerase. It's going to come back through, and it's going to replace it with DNA nucleotides instead of RNA. So these just served as a temporary placeholder. Once that happens, DNA polymerase also proofreads for any mistakes that it made, just to make sure the system is efficient.